Yes. Okay. Yes. Okay. Okay. So I'm Ms. Eiler, and this is my microscope. And today I have for you a variety of things to look at. So, yeah, this is what we're gonna do. So basically, you can see, oh, this big white thing in the back is the microscope. So you can see me in the bottom corner, but you can't see. It should zoom up. So is it not working, the microscope camera? Where is it? I can't. Let me get it up to somewhere. Oh, it's mad at me. This is an adventure. Okay, yeah, so part of this was I wanted to show kind of how microscopes work. So when you're using a microscope, you always start with the stage all the way down and nothing is focused, and you start on the lowest possible like magnification, and then you bring it up into as focused as you can get. And this is a little bit hard because what you guys see in the camera is not as good as what I see in... Let me see what I can get here so I can look, because I want to see what you're typing, but I can't see in the YouTube viewer as well as, there we go. So here we go. We're seeing some cells. So the first thing we're going to look at today is actually the mitosis slide. So if you're in biology class with us, hopefully you remember mitosis. So we can move it over a little bit too, so you can get a little bit better view. And then if we want to go a little deeper, we'll move it over to our next slide, and that's going to make it fuzzy again. So we have to bring it back into view. We can see, so these cells are going to all be in different stages of mitosis. So this is an onion root tip cell, and the tip of an onion root is growing. So all of the different cells are going to be going through mitosis. So then let's see if we can go one more. Oop, a little bit blurry there. All right, there, let's see what we can see. Hi, Sophie. All right, so here we go. We've got some, you can see some chromosomes around. Hopefully those are, you're remembering those from our genetics unit too. You can see them, if you're seeing them condensed like that, that's a cell that's getting ready for or in mitosis. Oops. And what's interesting about it is that when it's actually in real life, it's not ever as clear as what we're used to seeing in the pictures. You know, like when we look at a cell in anaphase, we're used to seeing them like those little A's out to the side, but it's really hard to find them like that in real life. Let's see if I can get this one. There's this cool one right here. Let's see if I can get that one out. It's hard. Your viewing screen is not as... There, that one is kind of neat. Kind of to the right of the screen is really... And what's cool too is you can kind of... They're 3D, so you can zoom kind of through the cell a little bit. You can see... You know, this is kind of the top, and then through the middle, and then through the bottom. So I just thought that one might be kind of interesting to us, because we had just done some mitosis. So let's pull up back out of there. Get ourselves reset. I also have, this one is a meiosis slide. So we'll do the same steps. We're back all the way down. And then we'll slide it up until it kind of comes into view. It's hard too because sometimes you'll see something, but it's actually just the glass from the slide. It's not what you're looking for. Okay, there's what we're looking at, but I think we just missed it. So you can't find the blood type in the meiosis and mitosis ones because it's not like detailed enough for us to be able to look at, but we are going to look at some blood slides. We won't be able to see the blood type directly in those either. You use a special test to do blood type like you do in the hyperdoc that we did in class. But that's a really good question. Okay, so here we've got, these cells are doing meiosis, so they're going to look pretty similar to our mitosis slides. And we're going to see some of those chromosomes condensing. Zoom in a little more. So this slide's interesting. You really, even less so than the other one, can't see like kind of those stages we talk about, but you can really see like those condensed chromosomes. You can see them. Oops. Like how dark that nucleus gets. And what they're doing in there. So remember, in meiosis, we end up with 4 cells at the end. See in there, you can see all those chromosomes. This microscope was my COVID-19 stress purchase. I wanted something fun to do with my students from afar. But I just think this slide is cool to look at in comparison to the mitosis one because it's, you know, it's a similar process. But oh, here we can see them separating. This is kind of this is an anaphase one, right here. You see them, you know, they're pulling apart from each other the chromosome. But yeah, I just think this slide is interesting because it's the same basic processes that are happening between meiosis and mitosis. But they look pretty separate from each other. Sophie, the test you do is you actually take some of the blood that you want to test and you mix it with the opposite types of blood and you see if it coagulates, if it clumps together, because the blood, it like, basically you can think of it like it kind of freaks out. It doesn't like to be with the wrong type of blood. And so if you mix it with the wrong types of blood, then it will clump together and like get all gross. So if you mix it with all the different types of blood, the ones that it doesn't do that with, that tells you, okay, this is the blood type. All right, so let's put away our meiosis. So actually I did pull out my set of slides, came with 3 types of blood, and like Sophie's asking about, we're doing blood typing in our genetics unit, so I thought it might be cool to look at some blood, and my set of prepared slides came with frog blood, fish blood, and human blood, and I thought those might be cool to look at under the microscope. So let's see what we got. So I've got frog blood first. All right. So it's over here. Okay. All right, yeah, there it is. So pull it up over there. And then we get it a little closer. Oh yeah, that's cool. Yeah. Okay, so these are some blood cells from a frog. This is really interesting. I've never actually looked at frog blood before, so you are experiencing this with me. Ooh. Getting a little out of focus there. It's interesting, it seems like they do have some of the other components of blood in here too, like some of the other types of cells. So it's important to know blood is not just one type of cell, there's lots of other stuff in there too. So that's some frog blood. It's getting out of focus again. I think it has been dyed, so usually, or stained. Usually on slides, we use different kinds of stains to make things like easier to look at. So this isn't actually what it would look like in the frog. It's just so that when we look at it under the microscope, it's easier to look at. Like when you look at bacteria on the slides, which we will probably do, if I keep doing live streams, we'll probably do that in a couple weeks. That bacteria aren't actually those colors. It's just we use different stains. Yeah, those are probably nuclei, which is interesting because human red blood cells don't have nuclei. So that's really an interesting thing to see there. All right, next we have fish blood. Let's see what's different about fish blood. Right. Bring it up. Okay, it's looking pretty similar from far away. Actually, looks a lot like the frog blood. There's a lot more cells on this one than on the other one. Ooh, why won't it focus? This is weird. That's a really good question. Most blood actually does appear red. Again, I think this is appearing red mostly because it's dyed. It's really weird. I can't get it into focus. Let me grab a wipe and see if it's just I touched my microscope. There's actually an animal called a horseshoe crab, and their blood is blue. Like when you, like if you cut them or whatever, their blood is blue. It's because it's based on copper instead of like iron or whatever. And it's actually really important. We use horseshoe crabs for a lot of really important medical uses. This is really weird. I can't get it to focus on this slide. We're not meant to see the fish blood. Okay, I can focus on that much. No, what in the world? This is my live stream technical difficulties, not being able to focus the microscope. Okay, now it's telling me things are disconnecting. Why is this camera? Okay, give me one moment for technical difficulties with the microscope camera. Remove. Oh God, that's a lot of me. Okay. Okay. Okay, I think I fixed it. Okay, so human blood is what we've got on deck now. Okay. Zoom in. Oh, that's too far. I do not understand why this is not focusing. There we go. This is the life of a scientist. Sometimes things, they just don't go the way you think they're gonna go. Okay, here we go. I see them, and then they escape. Okay, well, for some reason, this is as far as we're gonna get looking at these. So these are our human blood cells. But like I was saying to Mr. Raff earlier, you can see these ones don't have a nucleus. So the frog ones we saw, they had those big nuclei in them, but human erythrocytes, they don't have a nucleus in them. They're the only cells we have in our body they don't have a nucleus. When they're being made, they have one, and then they actually get taken out, which is a really cool process. I don't know why I'm not able to zoom in. I think there's something wrong with the lens on my doodad over here. Technical difficulties. Okay. My next three, my last series for this, I think, since we're having technical difficulties, is wings. Okay, everybody likes to look at wings. They're one of the cooler things to look at under a microscope. So I brought with me, all the way from my desk, dragonfly, bee, and butterfly wings. So hopefully they are cool to look at under here. So let's turn the light down a little bit. All right, let's see what we have. So this one is a dragonfly. That's glass. Oh yeah, here we go. Isn't that cool? Sophie, white blood cells in our body are there to attack infections. So all the different, that's what I was talking about earlier, there's a lot of different things in our blood. White blood cells are it's a broad category name for a lot of different blood cells that help your immune system. And they do a lot of different types of things. But primarily what they're doing is noticing things that shouldn't be in you, and they attack them. So this is our dragonfly wing. You can see kind of like these little cells basically shape. It's kind of neat. It's not like a very regular shape. Oh, it go a long way without... Oh, it's because I left the wing by accident. There it is. I kind of expected them to be like hexagons or something, but they're not very like a regular shape. It's just kind of like random. It's kind of like when you fly over, like when you fly out of a city at night, and like you can see all of the, I don't know, the way the city's like in weird shapes. Except that this is not as regular. That's neat. At the Science Museum, you can look at all sorts of different types of wings under microscopes. They have a microscope section, and they have like a whole bunch of wings that you can look at. All right, next wing. Let's look at a bee wing next. I love bees. They are super cool, and I want to save bees. Also, I really want to have like a beehive. Once I started a bee garden, like where you have special flowers that bees like. Oh, this is cool because it's kind of yellow. And it has like fuzz on it. Check that out. I did not realize bee wings. I wonder what kind of bee this is from. It doesn't say, it just says bee wings. It would be interesting to know what species of bee this comes from. Genetics is not our last unit. Genetics we will be doing for a couple more weeks, and then we will do microorganisms. So that's going to be like viruses, bacteria, fungus, and protists, and then we will do just a little bit of plants to wrap up the school year. I am obsessed with this bee wing. Oh, look at the edge here. It's like, it almost looks soft. Yeah, bees can be really dangerous, especially if you have an allergy. Miss Henry's allergic to bees. She has a sticker on her water bottle that says, like, be aware. And it's meant to be, like, be aware that bees are important, but I don't know, she thinks of it as, like, be aware that bees are dangerous. This is neat. I like how I really am obsessed with these little, like, kind of spiky bee thorns on its wing. This is super cool. Okay, last wing. Our last wing, butterfly wings. All right, let's see. Let's put it over here. Oh, this is not at all what I expected. There we go. It's kind of like, it kind of looks like feathers. Maybe like a moth or something. I think most people get honey from like the grocery store. Only certain bees actually make honey, which is really interesting. A lot of people think like all bees make honey. Also, individual bees make barely any honey, which is something I did not know but learned recently. At the Science Museum, the butterfly wing I looked at was like much bigger and more colorful. So this is a very different type of butterfly wing than I'm used to. It does look very soft. It's a much different butterfly wing than I'm used to looking at under a microscope. Usually when they say butterfly wing, they give you one that's like kind of what, I don't know, people think of as being a butterfly. I really like this one. It's a very interesting color. Well, I hope this was enjoyable. I'm sorry we had some technical difficulties. I'm not really sure what's going on with my 40 times magnifier, so yes, these wings are dead. Anything that's mounted on a prepared slide has been, has died. Unless you've been able to, I guess unless you've been able to remove it without it dying. But my best guess would be that, yeah, this is from a deceased organism. But yeah, so this is the end of my live stream. If you enjoyed it, let me know. Thanks, Shelly. I probably will do this again. I got a whole box, and I have a lot that are going to be relevant to what we're doing in our future units. So I enjoyed it. I thought it was fun. So thanks, guys.